opportunity to introduce my work titled ALKANNA ORIENTALIS Plant Cultures as Novel Source with Antibacterial Activity. This work actually is in the junction of three different scientific disciplines, the microbiology, plant physiology, and plant biotechnology. I hope it will be interesting for you. The object of my investigation is ALKANNA ORIENTALIS perennial herbaceous plant of Boraginaceae family, which growth area is represented here, and it is rarely found in dry and mountainous places at all regions of Armenia. The two other species of this genus, ALKANNA malatiana and ALKANNA tinctoria, are also represented in Armenian flora, but not as popular as ALKANNA ORIENTALIS in folk medicine and other area of human activity. The root of this plant is red-purple because of its cork layers contain red pigment chiconin and its derivatives, which have been used as a precious dye since ancient times. The already studied sides of these investigations are, in recent years, the preparations based on biologically active compounds of plant origin possessing antimicrobial and antiviral activity have been widely used for the treatment of various infection diseases. Second, some biologically active compounds of different plants, including ALKANNA subspecies, possessing detoxifying properties by means of inactivating the microtoxins and have antioxidant and cytotoxic effects. ALKANNA subspecies produce naphthoquinones known to have antimicrobial and cytotoxic activities. Pigment chiconins and its esters are the widely studied class of these substances, which are used in medicine, cosmetics, and food industry, and they are commercially very important. Plant origin quinones may suppress the activity of electron transport systems of many microorganisms and ATP production. Sarotrin from ALKANNA ORIENTALIS as an antimicrobial agent can inhibit efflux pumps activity of Staphylococcus aureus, and there is a little very scarce information concerning isolated cultures of Boraginaceae family plant, but isolated culture of ALKANNA ORIENTALIS as a source of naphthoquinones had not been examined yet. So obtaining of isolated cultures of ALKANNA ORIENTALIS, developing conditions for their cultivations, as well as maintenance of a stable growth and active metabolism are of considerable interest to develop new cell line processing, the ability to accumulate naphthoquinones. The aim of the present work was first obtaining of isolated cultures of ALKANNA ORIENTALIS, developing the conditions for their cultivation and maintenance of a stable growth and active metabolism, and investigation of antimicrobial activity of it, extracts by revealing the responsible mechanisms. Some methods have been used for isolation of in vitro cultures of ALKANNA ORIENTALIS. This plant was collected from the Sunik region in south of Armenia during the blossoming period. Approximate elevation of the collection place was 1,100 meters above sea level. The identification of plant was carried out in the Institute of Botany of the National Academy of Science of Armenia, and this plant was included in the same institute herbarium. The callus cultures were obtained using MS mineral base and vitamins containing medium, conditionally named number 1 control medium, supplemented with 2 milligram per liter BAP, benzyl aminopurine, and 0.5 milligram per liter EAA, indole acetic acid. further stable growth of callus tissue supported in two other nutrient mediums named medium number 2 and number 3, which differs with their compositions. But medium number 2, like medium number 1, but did not contain ammonium nitrate, and the copper concentration was increased in this medium to 0.2 milligram per liter. Previous concentration was 0.02 milligram per liter. Medium number 2 3 is like medium number 2, but only concentration of BAP increased to 3.0 milligram per liter. For further analysis, intact root and callus alcohol extracts were prepared. For that 1 gram of dried powdered material was homogenized in ethanol, and obtained mass was centrifuged, and supernatant was dried and used for further quantitative and qualitative analysis. Quantitative and qualitative analysis was performed by HPLC method, and antimicrobial analysis was performed using two different methods, agar diffusion and broth dilution methods. For this test, different gram-positive and gram-negative bacteria were used, as well as pathogenic bacteria such as Vibrio cholera, Yersinia enterocolictica, and Bacillus anthrax. Ampicillin was used as the positive control. Further analyses were done for detection of MIC of extract using dilution method. As a positive control is in this case, chiconin and ampicillin were used. For the determination of action mechanism of the extracts on bacterial cells, membrane vesicles were isolated from lysozyme treated Enterococcus hera bacterial cells using the buffer-lacking potassium ion. The strain was kindly provided by Professor Kobayashi from the Graduate School of Pharmaceutical Sciences of Chiba University, Japan. Obtained membrane vesicles were used to determination of ion fluxes across the cell membrane, accessible gel groups number, and third, ATPase activity. Ion fluxes across the cell membrane determined by the changes in proton and potassium external activities using appropriate selective electrodes. The electrode rangings were calibrated by titration of the same medium, and ion fluxes were expressed in nanomole per minute per number of cells in a unit of volume. Accessible thiol groups of membrane vesicles were determined by Ellman's reagent using spectrophotometric method, and glutathione was used as a standard. ATPase activity was measured by the amount of liberated inorganic phosphate after adding 5 millimole ATP by the method of Taussky and Shor. Inorganic phosphate was determined spectrophotometrically, and the ATPase activity was expressed in nanomole inorganic phosphate per minute and milligram protein. Whole cell and membrane vesicles were pre-incubated with ALKANNA orientalis extracts with different concentration, and were calculated this concentration based on minimal inhibitory concentrations of these extracts. At first, I want to mention that according to HPLC and spectrophotometric analysis, ALKANNA orientalis callus cultures of root origin only possess the ability to synthesize chiconin, and they produced also the highest concentration of naphthoquinones that were not shown here. The concentration of naphthoquinones in callus cultures of leaf and stem origin on medium 2 were lower, but under the influence of the medium 3 composition, their concentration increased. And on this basis of this data, subsequent studies were used using callus cultures of root origin. The inhibitory effects of ALKANNA orientalis callus extracts against various microorganisms were showed here. Gram-positive bacteria, including Enterococcus hera, I don't know, including where is Enterococcus hera, were the most sensitive to the substances of these extracts. Gram-negative bacteria were even less sensitive to the extracts. Note that biological active compounds synthesized in an isolated culture possess greater antibacterial activity than those of intact plants. The high inhibiting effects of the extracts were also detected against some pathogenic bacteria, but not significant effect were between the gram-positive or gram-negative ones. To determine the nature of alkanate tissues, biologically active compound action, their bacteriostatic effects on the test microorganism were established, and it was established that only in case of lactic acid bacteria they have bactericide activity. The positive control with ampicillin showed only approximately twofold higher activity in comparison with the examined extracts, and the negative control with ethanol showed no activity. Moreover, the results indicated that composition of the nutrient medium exerted a significant influence of the synthesis of naphthoquinones in the isolated alkanate cultures, and it was found that the elimination of ammonium nitrate from the medium, an increase of extract antimicrobial activity compared with the control, and their growth absence zones were larger significantly. After the revealing of the nature of antimicrobial activity of alkanate extracts, it is necessary to reveal mix against different bacteria. Mix of extract was established using dilution method. The latter helped to avoid the disparity which occurred when relative antimicrobial potency was determined by the comparison with inhibition zone sizes around extracts. Indeed, the absorbance of bacterial suspension was not changed at 2050 micrograms per milliliter concentration of extracts against Enterococcus hera. So we can indicate that in case of, in this case, MIC corresponded to the 2050 micrograms per milliliter concentration. MIC of chikunins as a positive control was approximately 31 microgram per milliliter. And it was revealed that long-time cultivation as well as storage in dried callus tissues did not significantly change the antimicrobial activity. In this slide, you can see the ALKANNA orientalis callus cultures of 16th subcultivation and 145th subcultivation, and yellow one is the positive control, and it was revealed that during the long-term cultivation, the antimicrobial activity did not change significantly. As it was mentioned above, the marked bacteriostatic effect was with gram-positive bacteria, including Enterococcus hera. In order to determine the action mechanism of this extract, some membrane-associated activity of hera was examined in the presence of ALKANNA orientalis intact root and callus tissues extracts. Indeed, the kinetics of proton secretion and potassium accumulation by HERA whole cells were determined when glucose was added into the same medium. Parallel experiments were done with DCCD. It is the specific inhibitor for FOF1 and other proton translocator mechanisms. Both protons and potassium ions fluxes were DCCD sensitive. There was a 2.6-fold inhibition of proton efflux in the presence of 0.2 millimole DCCD, suggesting the participation of FOF1 in the proton secretion. Different concentrations of ALKANNA orientalis intact root and callus tissue extracts influence on proton and potassium fluxes rate were investigated. Bacteria were treated by plant extracts for 10 minutes. The effect had a concentration-dependent manner. 10 and 20 microgram per milliliter extra intact root extract inhibited proton efflux by 1.26 and 1.46 fold, and potassium efflux by 1.25 and 1.36 folds respectively. Callus tissue extracts effect on ion fluxes was stronger. 10 and 20 microliter per milliliter callus tissues extract suppressed proton efflux by 1.54 and 2 folds, and potassium efflux by 2.8 and 3.8 fold respectively. In the assays, the CCD in 0. millimole concentration was added, and 5 minutes later, the CCD sensitive fluxes were calculated. Intact root extracts inhibited the sensitive proton flux by 1.16 and 1.5 folds respectively. 10 microliter per milliliter callus tissue extracts inhibited fluxes by 2.6 folds, while 20 microliter per milliliter callus tissue extracts inhibited almost all of bacterial growth. This result pointed out that ALKANNA orientalis intact root and callus tissue extracts affected of FOF1 enzyme activity. Membrane-associated ATPase activity of HERA was determined to be potassium-dependent and markedly inhibited by DCCD. The interaction of FOF1 with potassium uptake system could be due to disulfide interchange. In fact, ATP in the 3 millimole concentration increased the number of accessible thiol groups in membrane vesicles. The level of this group was 1.2-fold higher. However, similar effects were not detected when DCCD was present in the thiol groups in the presence of ATP or not. This result indicated that the changes in the number of thiol groups in the presence of ATP might be associated with FOF1. The HERA membrane vesicles number of thiol groups was determined in the presence of 20 microgram per milliliter ALKANNA orientalis tissues extracts as well. In addition, the addition of extract into the same medium decreased the number of these groups and lowered significantly. The strongest effects were obtained with 0.2 millimole DCCD. All these above mentioned findings could be significant for uses this plant extract as an alternative antimicrobial agent in pharmaceutical and food industry as well as in medicine. And special thanks to these people for preparing the work, and work was supported by the basic research support of our Ministry of Education and Science. Thank you.
Thank you very much. We have still time for one question from the audience. Yes, here.
What is the physical properties of this extract?
You may use the microphone.
What is the physical properties of this extract, And is it safe to use as food preservative or no?
Physical, what do you mean saying the physical? Smell. It is colored, red, purple colored, dried powder, dry powder, red colored, and we can use in food preservation for meat preservation particularly. What? May use. May use. Yes. Yes, it is safe.
According to FDA or according to FDA, recommended to add it?
No. We just investigated it and we just study, investigate, yes. Thank you too.
Do you know what is responsible for the activity?
What is the
What
response?
substance is responsible for the activity in the
The extracts contain chiconin, which have antibacterial, strong antibacterial effect. Chiconin and its derivatives. It's a red pigment. Yes, it is known since times.
Well, thank you. Here I'm him. Excuse me.
What? I don't hear you. Sorry.
Compare the activity of extracts from callus and from the mature plant tissues. I mean, what was the rationale? Was really the extract composition was different?
Yes, callus culture was more active. It is due to the medium components of these cultivation conditions.
Okay, and the compound you mentioned is not present in the mature plant tissues.
What? Sorry?
I mean, the active compounds you mentioned in the callus is usually not present in the plant tissues.
It is present only in callus cultures of root origin, not leaf or stem origin.
Thank you.
Just the last one.
I just want to ask, what's the essence of isolating A. orientalis from the culture instead of direct aqueous extraction of the plant? Then secondly, take into consideration McFarland method of standardizing your isolate before subjecting it to antimicrobial activity. Thank you.
Thank you for the questions. We isolate these cultures using well-known methods, using immense nutrient medium, and only change nutrient medium composition in order to have in mind the direction of further direction of our investigations.
Thank you very much.